for attending. Thanks. And thank you, Dr. Albensi, for providing the opportunity. My name is Eftekharpour. So I usually start with a joke with it. So this is basically John Johnson in English. So anyway, I'm going to be talking, today, some concept on neuronal nuclear damage in Alzheimer's disease. I'm going to be talking about nuclear lamina and laminopathy, which is a relatively new feature in Alzheimer. And also we're going to talk about the causes and consequences of nuclear damage. In this context, I'm going to be providing some evidence from the literature and also some of the unpublished data from my own lab. We're going to talk about tauopathy, a little bit of amyloid toxicity. We are going to be focusing mostly on oxidative stress, which is the major focus of my research, and specifically on thyroredoxin protein that I like to highlight here. And we have developed a new animal model linking thyroredoxin deficiency to nuclear damage and neurodegeneration. So hopefully you will be as excited as I am here. So first, here I want to just show what I'm talking about when I'm talking about nuclear damage. This is human tissue stained with lamin B1. Lamin B1 is a component of nuclear lamina in neurons. We have them in other cells, but is based on deletion study. We know that lamin B1 is one of the major protein that is involved in gene regulation. It's important for the resistance of the neurons to oxidative stress, especially in adult neurons. So in a normal tissue, when we stain for lamin B1, we can see that neurons have a very smooth expression of lamin B1 right under the nuclear envelope inside the nucleus. Just to give you an approximate localization, so nuclear lamin B1 would be just sitting between chromatin and nuclear envelope, so basically providing a glue function that binds to the chromatin and holds the chromatin in place. Specifically, a heterochromatin, which is the compact protein, compact part of the DNA, is associated with lamin B1. And what we see after in deceased patients, about 65% of the neurons have invagination of nuclear lamina inside, and they look like kind of coffee beans. These data have been recently published. So the first report for the induction of nuclear pathology perhaps came in about 10 years ago from Dr. Feeney's lab and her graduate student, Bess Gross, which now is a professor at Houston. I think it's Anderson Medical Center there. She has a very interesting series of discoveries on nuclear lamina damage. We independently came to this and we learned about her research, and I will describe later on. But here is a diagram showing that in their discovery, they show that tau hyperphosphorylation basically causes changes in the cytoskeleton, causing actin fibrillization, and actin is kind of pushing down on the nuclear envelope, causing invagination of the nuclear envelope into the chromatin. A series of events happens that I'd like to summarize here, and then these causes long-term changes in gene expression and function of the neuron, which eventually will actually compromise the neuron survival and neuronal loss. So what we see here, this is nuclear lamina. First of all, let's just review your cell biology. When I learned about nuclear lamina, this was just recently, and in my undergraduate last century, we never learned about it. So here is where nuclear lamina is actually located. This is, as I mentioned, close with the interior layer of the nuclear envelope. And this is the chromatin. And chromatin is actually binding very closely and intimately with lamin to the endoplasmic reticulum and nuclear envelope and moves with that. When nuclear damage happens, either one layer or both layers of the nuclear envelope actually protrudes into the chromatin. And this changes a lot of things. One of the factors that is highlighted here is a nuclear calcium, that in the nucleus we have a depletion of the nuclear calcium, and then also there is interruption of nucleocytoplasmic transport. There is an accumulation of RNA at the sites. As I mentioned, nuclear, heterochromatin is bind with lamins. This invagination also causes decondensation of heterochromatin and expression of new genes. So the component of the nuclear lamina is basically three genes produce nuclear lamina. Lamin AC is the alternate splicing of one gene. And these guys are very commonly related to laminopathy in other tissues other than central nervous system. There are about 300 mutations, mostly in the mesenchymal tissue. These are related to, for example, the progeria disease that you know of. We have the muscular dystrophy, lipidostrophy, cardiomyopathy, and Hutchinson-Gilford progeria. These guys are flagship diseases that is caused by mutation in lamin AC. In the brain, we don't have a lot of lamin A. It's mostly lamin C. Lamin A is downregulated in the brain through a microRNA regulated system. We're not going to be talking more about that one. But lamin B1 and B2, both of these are two different genes. They are just recently, we don't know any mutation in these genes that is related to any of the diseases that we know of, but we know that there are damages, structural damages that occurs, and most recently, Alzheimer's disease and other tauopathies are known to be associated with damage to nuclear lamina and disruption of lamin B1. So a series of studies here, I have cited them, started with, these are mostly what I mentioned, Bess Frost in Texas. Her research started in Drosophila, and she showed that in a model of tau hyperphosphorylation, there is nuclear lamina invagination. This caused chromatin relaxation, and this was associated with neuronal death. And what they did, they prevented nuclear condensation using pharmacogenetics approaches, and they showed that decondensation of the chromatin is actually lethal, but could be prevented. They also used they showed that oxidative stress was sufficient to induce and drive heterochromatin decondensation. They have two models of Drosophila. One was thyroidoxine reductase deficiency, the other one superoxide dismutase. And what they did, they also showed that nuclear damage is also happening here. And they blamed tauopathy that is responsible for changes in actin fibrillization, bundleization of actin, and putting pressure on nuclear damage. Then what they further looked at in these models, they looked at RNA sequencing, and they showed that tauopathy in Drosophila was associated with aberrant activation of retrotransposons and reemergence of the ancient retroviruses. This was very interesting, showing that they showed that protective mechanism in human genome that are usually active and prevent retrotransposon activation are downregulated, partly perhaps due to decrease in calcium in the nucleus, and this causes activation of retroviruses. So they also looked at human samples, and using RNA sequencing, they showed that these ancient retroviruses and retrotransposon activation does occur in human tissue. So this has been followed up. BESPROS has shown that calorie restriction positively decreases retroviral expression in Drosophila. So this is really, something good news, that changing in our, life and positive, changes in our life could actually affect our long-term survival and brain health. And also now she is focusing on clinical trials using antiviral drugs to see if this would be able to prevent dementia. And these are currently in progress. So basically that was a nutshell what happens at the level of the cells, whatever it is. Instead of looking at amyloid beta or tau, let's see what happens in the nucleus, and are these events preventable or not. So now I want to jump and look at the oxidative stress. So we know that oxidative stress is very well related to the pathology of Alzheimer's disease, and this is how we ran into the nuclear lamina damage. So my lab is interested in thyroidexin. You all know, just reviewing here, thyroidexin is a small protein, about 12 kilodalton, and has 2 thiols in its active site. These thiols have very low redox potential, meaning they can easily donate their electrons to the substrate. Substrate could be hydrogen peroxide and free radicals that they are being used to control the level of hydrogen peroxide. Thyroidexin is also very important for peroxyredoxins. Peroxyredoxins, are important for measuring, regulating the hydrogen peroxide level, and a lot of signaling system and kinase activation is being controlled by peroxyredoxin. All of these systems will be affected when thyroidexin is oxidized. And oxidized thyroidexin has to be again reduced by thyroidexin reductase using electron from NADPH. So, and what we know is that there is, thyroidexin is a really good electron donor to oxidized protein. And there are plenty of proteins inside the cells that are susceptible to oxidative stress. We know that about 240,000 cysteine residues are found in human proteome. That is really a jump in the content comparing it with lower vertebrates and in bacteria. About only 10% of these guys, these cysteine residues, are accessible to ROS, and they can be involved in cell signaling. So they will be oxidized, and then thyroidexin, having a low redox potential, can go and rescue these proteins from further oxidation. And to my knowledge, thyroidexin is the only one that does that. Glutathione will be able to go and partially reduce these proteins, but will be just binding to the protein and will be making a glutathione adducts. But thyroidexin, in a very quick transient reaction, will donate electrons to the proteins, will reduce the substrate, and then will go on for reduction by thyroidexin reductase. And the level of thyroidexin is very low. It's about 5 to 10 micromolar compared to 10 millimolar for glutathione. And also the other thing that we know in a neurodegeneration and lack of these guys, thyroidexin 1 and thyroidexin 2, which is mostly mitochondria, is embryonically lethal. In CNS diseases, and there is a lot of information that is related to thyroidexin. What we know is that in Alzheimer's disease specifically, there is depletion of thyroidexin. in the brain samples. We also have shown that one is in also Western blotting in our own hand. We also know that there are evidence of decrease of thyroidexin system in Parkinson's disease. Mutation in thyroidexin reductase has been found in ALS, and these are mutations that will render thyroidexin reductase inactive, and therefore it would be causing oxidation of thyroidexin and also its low level. We know that thyroidexin reductase is decreased in Parkinson's patients. And to make everything worse, we have an inhibitor of thyroidexin, which is thyroidexin inhibiting protein. This protein comes and binds to thyroidexin active site and renders it inactive. We know that TXNIP is upregulated in Alzheimer's disease and also in Parkinson's disease, and this will enhance the oxidative stress. So everything here, in my books, points out to thyroidexin. So my hypothesis is that whether depletion of thyroidexin system in neuron is sufficient for induction of neurodegenerative pathology. So for that, we have been working on this for a while using in vitro cell models. This is a model of SHSY5Y neuroblastoma cells. What we did, we used pharmacological and genetic inhibition of thyroidexin. We used inhibitor of thyroidexin, PX12, and also thyroidexin reductase. And what we showed, focusing on autophagy and apoptosis, we showed a decrease in lysosomal activity. Here is the cells in control, and the red dots are supposed to be showing active lysosomes. With serum deprivation, as you know, there is an increase in lysosomal activity, as we can see here with increased red dots. When we inhibited thyroidexin reductase with auranofin, the level was very low. And here is looking at electron microscopy, and we see that there is an accumulation of autophagosomes that are actually just accumulated inside this, the cytoplasm, indicating a decrease in lysosomal activity. So, and we know that autophagy is also decreasing in Alzheimer's disease. So this is 1-for-1, thyroidexin is depleted in Alzheimer's disease, and it's perhaps related to decreased autophagy. So we have shown those. Then we started looking, my postdoc came to me and he said, we have nuclear damage. And I said, so what? And he says, no, we have nuclear lamina damage. And I said, what is nuclear lamina? So he showed me this, and we were looking at these. So here we are looking at lamin B staining in SHY cells, and we see that in normal control condition, lamin B1 is located very nicely around the nuclei and some inside the nuclei. And then when we put the cells under serum deprivation, cells start starving, nucleus is starting shrinking. Very little, if any, invagination could happen here. But when we inhibited with PX12, which is an irreversible inhibitor of thyroidexin, we saw that these cells, these nuclei, were actually crumbled. And this was associated with caspase-6 activation. So, and here it is. My postdoc was working on nuclear lamina, so I was lucky that he knew what was going on, and it was just everything confirmatory. He ran a Western blot, and he observed a 46 kilodalton band. He said this is a signature for caspase-6. And then here we are quantifying this. We show that there is an upregulation of the cleaved lamin B. And here is the activation of caspases. Here we have shown it with only Western blot, but also we have confirmatory results with enzymatic activity, and we can see an increase in caspase-6. This is the caspase-6 activity that I was talking about. So interestingly, when we pre-treated cells with a caspase-6 inhibitor, we were able to actually inhibit nuclear lamina invagination, and the cells were completely normal. There was a good decrease in the amount of cathepsin, caspase-6 activity and damage in nuclear lamina. So here, we asked whether damage to nuclear lamina is thyroidoxin specific, or anything that causing oxidative stress will be able to actually show that. In this study, we used a different sets of inhibitors for thiol system. So we used PX12, which is a thyroidoxin inhibitor, a specific inhibitor. CDNB is a thiol inhibitor in both of them for thyroidoxin and also glutathione. And then here we use BSO to deplete the reduced glutathione inside the cells, and aronothine, which is an inhibitor of thyroidoxin reductase. And what we show here is that inhibition of glutathione was not able to actually, prevent nuclear lamina damage. So this means that glutathione was not involved in nuclear lamina damage, and it's mostly inhibition of thyroidoxin system, which is responsible for that. So these are the quantification for procaspase level, and these are also for nuclear lamina damage. So now we looked at, we went to our neighbor, Ben Albensi, when he was in Manitoba, and I said, we definitely need your mice. And he gave us his 3XTG mice very generously. So we looked at these mice to see if our, hypothesis on thyroidoxin and lamin damage is true there. So here we are looking at different factors here. So here I'm showing you the thyroidoxin level. So we're looking at 2 months and 6 months old mice, and we see that there is a decreased thyroidoxin level in these animals. This is associated with changes in caspase-6 activity, and we can see the decrease in procaspase level, meaning increase in caspase-6 activity. And we also see that there is a correlation with increased lamin B damage. Here, what I have shown here in blue box, we are looking at the 46 kilodalton lamin B damage, and that is associated with decreased thyroidoxin level. So all of this was good, that, okay, so this mouse model is actually cooperative and shows us what it is. But we also observed this, a new band, a lamin B positive band, which was 21 kilodalton. And this was specific. We have used different antibodies, and we know that both of these pieces are C-terminal bands. We have, in this paper that was just recently published, we have used purified proteins, and we have looked at, pattern of cleavage between caspase-6 and cathepsin L. And we see that, they are completely two different proteins, but both of them working on lamin B1. So we don't know the importance of this, but, basically this is what we are seeing. So in this model, what it shows is that the depletion in thyroidoxin system is associated with increase in lamin B, level and cleavage of lamin B. So through a bunch of studies, we finally figured out what was the cause for lamin B 21 kilodalton, and we learned that this is cathepsin L activity and not cathepsin B activity. So cathepsin L was completely able to actually do that and was preventing. So here is what I showed you in nuclear lamina in human tissue. But here we then stained our nuclear or human tissue for lysosomes and for cathepsin L. And here what we are showing is that there is an upregulation of cathepsin L activity, presence here. What we can see here, huge upregulation of cathepsin L in AD patients. both we can see here, looking at this 3D, we try to show the location of these lysosome or cathepsin L positive lysosomes inside the cells. And we can see here a few things that we can learn from here. First of all, you can see that there is much more lysosome cathepsin L in AD patient, and they are also more closely related to the nucleus. And then also the other thing that you can see, the density of the nuclei and chromatin here, staying with DAPI, we can see that in AD patient, there is a condensation and compaction of chromatin in these patients that are obviously, they are deceased. So this perhaps would be a late stage activation of cathepsin L. Maybe this is what is causing at the very late stage. One thing that I forgot to highlight about caspase-6 is that caspase-6 apparently is an early biomarker in Alzheimer's disease. And there is a group in Montreal that they have shown inhibition of caspase-6 could be a mechanism, could be preventing dementia in a mouse model. So that's another thing that is, putting everything in mechanistic link, thyrodoxine depletion, then caspase-6 activation, nuclear lamina damage, and so on, towards the end, and neuronal loss. This is human tissue. Looking at all of these, we can identify the 21 kilodalton band that we see, but we could not find caspase-6 activation. But we could see caspase-6 activity and lamin B damage in mouse, but not in human postmortem tissue. So the question that we asked, whether caspase-6 is a substrate of cathepsin L, and at the very late stages, perhaps cathepsin L is taking over and cleaves caspase-6. And we confirmed that in vitro and with purified protein, that yes, perhaps at the later stage when cathepsin L is activated, it will degrade everything, including caspase-6. And that's the reason we won't see that in a human patient. So so that is one model. So we can see caspase activation, cathepsin activation, nuclear lamina damage. So here we also are showing here some of the cultures, primary cultures in cortical neuron. Here we are using an amyloid beta toxicity to show whether nuclear lamina damage is also present in amyloid beta toxicity. Amyloid beta is added to the cells, and then we look at nuclear lamina damage. We can see huge, invagination of nuclear lamina, and using cathepsin L inhibitor, we are able to actually prevent that and decrease that significantly. We are also showing that with amyloid beta, there is an upregulation of cathepsin L activity and damage of nuclear lamina B. So that's second model. So nuclear lamina damage is also is in amyloid, is seen as amyloid beta toxicity. The other model that people have shown is the tauopathy. So here is again a model of tauopathy in SHSY5Y cells, and what we have done, Parisa has upregulated, overexpressed actually tau here, full tau and pathologic tau is being upregulated here. And what we can see here, we are staining the actin and looking at the cytoskeleton. We can see this is normal cytoskeleton and nuclear lamina B in control cells with presence of full-length tau. There is much more nuclear invagination, as well as condensation of the cytoskeleton. Here is the C-terminal part of the pathologic tau, and we can see a huge amount of actin fibrillization. We have been quantifying these, but I'm not showing it here right now. But we think that with presence of pathologic tau, there is increase in nuclear invagination and also actin fibrillization. So we went on, and also on top of the neuronal model and using PX12, we used a genetic model, and Shakila here has shown that inhibition of thyroidexin using thyroidexin siRNA is able to induce nuclear lamina invagination. Again, looking at the cytoskeleton, changes in the cytoskeleton is occurring, but the good news is that we can prevent that very significantly using thyroidexin mimicking peptide. CB3, these are very small peptide, 3 amino acids that they are relatively stable for 24 hours. You can add them into the culture. People have used it in animal models and also in cell culture, and they are showing that they actually are able to functionally replace thyroidexin, and we see that in our own hand that there is a huge decrease in nuclear invagination and also normalization of the cytoskeleton. These data are currently unpublished and are going to be submitted hopefully soon. The other thing that Shakila showed is that, okay, so what happens inside the nuclei? So after induction of nuclear lamina damage, we also ask whether this can actually changes anything in the DNA. And we use gamma H2X, which is basically a marker of double-strand DNA damage. We see that induction, downregulation of thyroidexin is sufficient to increase amount of DNA damage, but this DNA damage can be treated and prevented significantly with addition of CB3 there. She has expanded these into primary cultures. Here we are inhibiting thyroidexin in primary cultures, and we can see that after inhibition of these cells, with about 2 weeks after culture, we can robustly see nuclear lamina damage in these cells. Here is gamma H2X that is being actually produced by downregulation. And more importantly, here we are looking at the synapse formation. We have stained for PSD95 and synaptophysin. And here, Shakila is looking at the co-localization of PSD95 and synaptophysin as markers for actually functioning synapses, hopefully, or structurally sound synapses. And based on our preliminary data, we see that there is a good decrease in the amount of synapses marker. So perhaps depletion of thyroidexin is related to decrease in synapse formation. Well, this could be related directly to thyroidexin or indirectly, because here what you are seeing, we are looking at the structure of the actin inside the cells and looking at the cytoskeleton. We definitely see basically fibrillization of actin in the cells. So changes in the synapses could be a secondary function of cytoskeleton changes. So we are not able to confirm that just yet. So now here, we're going to just jump into the animal model. At the end, I want to show you this. So using Curie recombinase technology, we generated a pan-neuronal thyroidexin depletion under the direction of the synapsin-1 in mouse. We see that these animals show a progressive depletion of thyroidexin. Here we have the 4 weeks and 8 weeks animals, and we can see a good decrease in thyroidexin level in the cortex of these animals. these animals, unfortunately, do not survive long enough, and between 8 to 10 weeks, they will die with a lot of problems that I'm going to be just showing you. So, and that's why we have opted out to sacrifice these animals at 8 weeks and look at their molecular changes. So first of all, at this age, starting around 4 weeks and higher, when we look at their hind limb clasping, we see that these animals significantly show that when they are picked by their tail, and this is their survival. Animals, do survive up to 10 to 12 weeks. But as I mentioned, just for the humane reasons, we sacrifice them around 8 weeks. We put them on rotor rod and also sensory motor test using tape removal. We see a significant, inability of these animals to basically, for their motor function, they cannot keep themselves on rotor rod, and also they cannot even, even feel the tape on their paws, so they won't be removing it. So far, based on what we see using BCMAS tests, unfortunately, they don't show any deficiency in BCMAS tests. So seems to be they are normal up to this age. But because these animals, do seizure and die, we are not able to, basically rule out the effect of thyroidexin on their memory. We also have looked at the size of these animals. These animals are smaller. The muscle mass is significantly lower in these animals, looking at their hind limbs and measuring the diameter of the femur and also on the shin. And we see a decrease, significant decrease in those animals. So here, we are showing a confirming depletion of thyroidexin in most of the CNS part. We can see here, and this is the dentate gyrus in the hippocampus. We have a good decrease in thyroidexin in these animals, and this is somatosensory cortex, and this is the lumbar cord. We can see that, so just basically showing that there is this model seems to be working, and there is a general decrease in thyroidexin. Interestingly, when we looked at the corpus callosum, we see that these animals are significantly deficient, and the thickness of the corpus callosum is roughly about 30% of the control. We also have this issue. We have looked at the white matter in spinal cord, and this is the dorsal part of the spinal cord, dorsal column. We see the myelinated axons are very thin and very rare. There is a significant number of the axon area, and also the number of axons is depleted in these animals. We also have done MRI recently. I don't have the data, but two areas that are significantly affected here is one is the size of the corpus callosum, and the other one I forgot. I have to. I just learned about this a couple of weeks ago. Anyway, so what we show here is that there seems to be a lot of degeneration or lack of, because this is a developmentally generated animal, or lack of development of the corpus callosum in these animals. Those are the kind of things that we have to still find out. Here we are looking at the nuclear lamina invagination and looking at the efficiency of this. Based on, I don't want to walk you through all of it, but what I can tell you is that except for hippocampus CA1, the rest of the spinal cord or somatosensory cortex or even dentate gyrus, they showed a good correlation of nuclear lamina invagination with thyroidexin depletion. But for some reason, CA1 was not showing. And incidentally, CA1, we used LTP formation in hippocampal slices, and we did not see any deficits in these animals. So there seems to be at least a negative correlation that we have there, but we still have to find out what's wrong in terms of memory and those kind of things. We also here what we are showing is a lot of some, not a lot, of neurodegeneration. We have stained spinal cord and cortex with the fluorogate, and we can see that some of the neurons are showing markers of neurodegeneration as labeled with this. This is an evidence of lack of synapse or a lack of efficient synapse formation in thyrodoxine neuronal knockout mice as tested by PSD95. The other markers for neurodegeneration, we should look at glial cells. Here we are showing you the dentate gyrus, and we can see that there is a good correlation and increase in astrocyte with thyrodoxine knockouts. But what we can see here, there was not changes in NG2, oligodendrocyte, but their morphology was different. So the number were not deficient, but looked like that these cells are not able to grab axons and myelinate, but there was a good decrease in the amount of, increase in amount of microglia and also GFAP, so indicating some form of neurodegeneration and glial response in these animals. So other markers of Alzheimer's disease also we tested using Western blot. So just to here is our thyrodoxine. These are 8 weeks animals in the hippocampus region. Over the whole hippocampus, we have homogenized and looked at. What we can see here, there is an increase cleaved lamin at 46 kilodalton, which is the caspase-6 signature. And we also have an increase in 21 kilodalton, which is cathepsin L signature. So this holds our hypothesis so far. The other thing that we can see here is, so this is what we see from here on these markers. So the other markers link, other evidence that we have is that lack of thyrodoxine is shown to be related to activation of Jun kinase and specific phosphorylation of tau phosphorylation in a specific residues, and here we are showing that we have 2 of the markers using 88 and also serine 396. We see significant upregulation of tau hyperphosphorylation that are immediately done by for some reason, my phone started talking. Then we see that the activation of Jun kinase is occurring in these animals, and it is associated with upregulation of the tau phosphorylation. So this is another marker, 8180, which seemingly, or from what we learned from the literature, may not be a direct effect by Jun kinase, and it's not significantly upregulated in these animals either. So the other markers that we have found, these animals also have increased amyloid beta. Total amyloid beta is increased in these animals, and also here we are showing that there is an association with neurons damaged with nuclear lamina with phosphorylated tau. Okay, another marker. This is TDP43. So TDP43, as you know, this is a DNA binding protein that is important in a neurodegenerative disease. So depletion of NDP in the nucleus seems to be a problem in ALS. It's toxic accumulation. Its accumulation in the cytoplasm is toxic. Here, Tatiana, she has done self-fractionation, looking at the cytoplasm and nucleus, and she shows that there is an accumulation of the PDP43 in the cytoplasm and depletion from the nucleus. this is, indicating that perhaps these animals may also show some evidence of ALS, but that remains to be understood. Further, we see that the neurons in the lumbar area are not really normal. They are, here we are looking at the motor neurons, looking at NF200, and we see that there is a huge accumulation of the NF200 in the cell body, and there seems to be less axons in these animals. So first of all, this perhaps shows that there is a decrease in axons, as we showed with immunohistochemistry, but also the other thing is this, indicates a deficient transport of the proteins into the axons. And this is something that we are looking at it, and we're trying to see if there is any changes in transport of the material. Anyway, so I am hoping that I haven't, make you dizzy with so many different angles that, I'd like to highlight here. Here, I'm going to just summarize. So this is this seeming to be a neuron. We have the nuclear envelope. Nuclear envelope is associated with lamin B1, and lamin B1 binds to the heterochromatin. For some reason, this could be increased oxidative stress and decreased thyroidopsin, increased tau phosphorylation, or changes in the cytoskeleton. Nuclear envelope starts invaginating inside the nucleus. We have a damage in nuclear lamin B1. Changes in chromatin has been reported from the dense chromatin to euchromatin. This could be mediated by caspase-6 and cathepsin L that we showed. The product of this would be decreased calcium and increased the ancient retroviruses from the cells. And also the other factor that is happening is a blockage of the cytoplasm and nucleoplasm transport of the material. Altogether here, this seems to be, contributing towards a development of neurodegeneration. And so what this perhaps would do this, our model, it shows that perhaps oxidative stress is sufficient to induce everything, including, as we showed, increased amyloid beta and tau phosphorylation and pathology that is related. So whatever the cause is, nuclear lamina damage seems to be, rational. It is rationalized to look into this system and see what's happening in the nucleus after this. Is it, long-term changes that occurs in the nucleus and genes related to the health of the neurons? We don't know. We have a bunch of different models that we are currently looking at, including looking at epigenetic changes that we have. We have done an epigenetic chip assay, and we are going through that one. The other model is in our knockout animal, which is a really interesting model. The other model that we have a thyroidoxin model that has a trap protein. This model is able to identify all the thyroidoxin protein targets, and that is something that is undergoing just recently with the proteomics study, and we are hoping that we will be able to actually produce those results soon. And then eventually we would like to be able to actually prevent all of these damages. Can we use those thyroidoxin mimicking peptide to prevent these changes? Anyway, these are people that have been helping with that. Some of those people I showed. I am, grateful to very little funding sources that I have, but a lot of collaborators that I have here at U of M and abroad. Ben used to be on this top category. Now I have demoted him to down there, and he is there, enjoying the warm weather. Thank you for your attention. I would be happy to answer any question you may have.
That was really great, Eftekar. Such beautiful images, and I think it's such important work. I'm just surprised sometimes that I don't see people talk about the nuclear aspects more at some of the big Alzheimer's meetings. But I do have a question for you while others are formulating their questions. Have you tested any of the classic Alzheimer's drugs yet, or even the two new drugs at all, with regard to looking at potential changes in the laminin and other aspects of the nucleolus in that?
So far we only in our tau pathway model, we are using some of the tau kinase inhibitors. I forgot the name right now. There's some bunch of numbers, but we are looking at see if inhibition of tau phosphorylation would actually be beneficial for the nuclear lamina changes. We do see some changes in nuclear lamina, but still those data are too preliminary to actually talk about it. We also have tried in these animals, trying to see if we can use any of these animals, but because the lifespan of these animals is so limited, we are really tied, our hands are tied. And the next set of animals that I'm hoping that I will have money to proceed is to target thyroidoxin deficiency into specific region of the body and not to the whole central nervous system. As I mentioned, epilepsy is just recently shown that mutation in thyroidoxin in the midbrain, which we do have it here, causes epilepsy in rats. And there are a couple of reports out there, and they are very recent data. So I think that what we are producing here is really a model of lethal epilepsy, and perhaps that's not a really good model for neurodegeneration. So my next step would be to go in and inject, because we have the thyroidoxin flux animal, we can go with Curie recombinase technology specifically to different region and see if that is able to actually induce neurodegeneration and Alzheimer.
Well, those comments are very interesting. And I don't know if you know this, but when I first moved to Manitoba many years ago, I worked with a different transgenic model. I worked with the presenilin-1 knock-in model that I got from my old boss, Dr. Mark Mattson at the NIH. And we routinely saw seizures in those Alzheimer's transgenic mice. And in fact, I've been to epilepsy meetings and I've been to Alzheimer's meetings, where both meetings have workshops on interesting connections between seizures, epilepsy, and Alzheimer's disease. So, you might really be onto something here. I just have one quick question, and I'll open the floor up to everyone. In the TRX-NKO, the knockout model that you described, have you looked at learning and memory parameters at all?
Yeah, only Y-maze, we have done some tests, and we see that they do the normal alteration. But the problem is that because these animals don't like to move, they don't do it as often as the other one. So, but regular alteration or spontaneous alteration happens, and they go 1, 2, 3, around the clock. I don't think they have that. We also have done, as I mentioned, LTP with Mike Jackson, and we were not able to show functional deficits, at least in CA1 neurons, which makes sense because we see that CA1, they don't have a lot of nuclear lamina damage, and also they have a good level of thyroidoxin left in them.
Okay, very good. So any questions from the audience?
Yeah, I have a question. I really enjoyed the presentation. I'm sorry that I was late getting back from class, so I missed the first part. But one of the things I'm curious about, what inhibitor of cathepsin L were you using? And is it selected for cathepsin L?
Yeah, let me see. To be honest, I have forgotten the name, but it was the one that was relatively better. We use this, one of them, let's see. Yeah, I know that a lot of these inhibitors are not really that good. Yeah, so Z5-CHO, we use that one, but we also have used genetic inhibitors. We use MEF cells lacking cathepsin L, and we were able to actually show that cells that they did not have cathepsin L, they were insensitive to amyloid beta.
Okay, and the other question I have is, was this a total knockdown of the thioredoxin? It looked like you had a couple of wisps on your Western blots. So is it partial that you, is it possible that you still had some partial thioredoxin function?
For we looked at Western blot, we looked at the tissue. As I mentioned, this is mostly neuronal. We would expect to see some thioredoxin left, and that would be the glial cells.
Okay. So you're not knocking out the glial cells.
No, we don't want to do that. We know that knocking out thioredoxin is completely lethal. So we're going to just look at the neurons and specifically focus on neuronal thioredoxin.
Okay, great. Thank you.
Eftekkhar, I also just wanted to mention quickly, you may know that I'm the editor-in-chief for a journal called Molecular Neurobiology, and we have a special issue going on right now. It just started. My guest editors are Brian Balin at the Philadelphia Osteo College or College of Osteopathy, and then Nikki, who's the principal and founder of the Intracell Research Group. Both of those individuals are interested in infectious agents and their links to Alzheimer's disease, and that is the topic of this special issue that they're the guest editors. And I say all this because of your comments in your slides on retroviruses and links to your model. So do you have any other comments on that?
No, that would be very interesting. We actually have looked deeper into this. And we have, evidence with real-time PCR that there is an upregulation of the retroviruses, in the cells. But we went one step closer, and we tried to do DNA methylation studies on the specific line 1 genes. And line 1 was, not. We could not identify significant changes in methylation of at least line 1. Again, we would need to do a global DNA methylation study to see what's going on.
Well, I'll have to introduce you to my colleague. In fact, we just published a paper in Alzheimer's to Dementia. I'm just a co-author, so I didn't do most of the work. But Nikki Schultz, like I said, she's the principal and founder of the Intracell Research Group, and they just published a new paper, and it's not even out yet, but it's been accepted on. And Rudolph Tanzi at Harvard is a co-author, and so is George Perry. That really kind of talks very seriously about this, controversial issue about infectious agents and Alzheimer's. So I'll have to introduce you to her, and you might be interested. Yeah. Okay, very good. Well, any other questions? We're running out of time very quickly. It's now 1 o'clock. I'll just remind everyone that I'll post a recording of today's talk with to my YouTube channel later today.
Yeah, I have one quick question. Yeah.
Go ahead.
Okay. So just what will happen, or have you seen when you induce autophagy or by caloric restriction, do you see any reversal of the pathology in the thyrodoxine deficient mice? And
we have not done that. We have not done that. These animals are very fragile, and they don't live long enough. And as I mentioned, those are huge studies that hopefully someday we will have money to do that. But that's a good idea.
Thank you. Yeah. Just also I have other quick question because when you in the thyrodoxine deficiency, you see the less survival, and also so whether have you seen the expression of the sirtuins like those are directly involved in, I mean, inducing survival. Whether do you see?
Very good questions. No, we have not looked at it, but you know, the list is getting longer and longer. You know, we really have a lot of materials to look at, and I'm hoping that this you know, will be the next stages. As you know, just the model has kind of disappointed me with the low survival rate. You know, if this was you know, at least 4 months, then we could have done a lot of things. You know.
Yes, yeah, definitely. And also you can check the proteins involved in heterochromatin regulation. So since you are seeing those changes, the remodeling of heterochromatin, so you can see the actual proteins, I mean, regulating the heterochromatin. So you can see those epigenetic changes also.
Yeah, yeah.
Okay.
Now, those are some of the studies. Hopefully you will see soon. You know, we have done those and we're just going through the data.
Okay, thank you. Yeah, great presentation. Okay.
Thank you. I appreciate that.
Thank you. Okay.
Well, thank you again, Eftekkhar. Very nice to see you. Thank you for a great presentation. Really beautiful images, and it's an important topic. And I'm just hoping that this starts to cascade, and you get that important funding to continue your work.
Thank you. I appreciate that. Thanks for the invitation again. And you know, so my name is very unique. And if anyone of the you know, attendees have any question, just reach out to me. You know, so my first name @umanitoba.ca. You know, it will show up.
Okay, great. Thank you. Okay,
Yeah,
goodbye,
thank you.
everyone.
Take
Bye-bye.
care. Have a good day, everyone.